Okay, welcome everybody. So we'll go ahead and get started. This webinar will cover release 45 of EuPathDB resources. My name is Omar Harb, and I'm part of the outreach team at EuPathDB. I'm also joined by Evelina Bashenko, who specializes in fungal organisms, and Suzanne Warrenfels, who covers all the parasite databases as well. So today's webinar will be recorded, and so if there's anything you missed or you would like to share with colleagues, you'll have access to the webinar recording, which will be, you should get a link from GoToWebinar, but also we'll post in our YouTube channel for others to see. Just a couple of technical points about GoToWebinar. If you have a question throughout the call, feel free to use the question or chat panel. You can type your question, and one of us will be monitoring those questions throughout, and we will either answer them right when they come up or afterwards or at some point during the webinar. The second thing you can do is raise your hand. So right next to your name, there's a raise hand option, and this will indicate that you have a question, and we can unmute you and allow you to speak if you prefer to voice your question by voice, or it'll just indicate that we should look maybe in the question section if you had typed a question. All right, so today, and everybody should be seeing my screen right now, and I'm showing you the homepage, the EuPathDB homepage. And one of the easiest ways to find out what is new in all of our resources is to go to the news section, which is right here on the left-hand side. And if you click on all news or all EuPathDB news, you will see that it's segmented into each of our databases. And as usual, not all of our databases get new data in each release. It just depends on what data sets we had on the docket and prioritization issues. But all databases are built on the same infrastructure, so any feature we load or make available for any of our databases will become available for everybody. So that's a nice feature. And the way we're going to be running this webinar is we will be tag teaming, and so I'll be switching back and forth between myself, Eve, and Suzanne. So to begin with, so there are a couple of things that we will be covering. One is we will highlight some of the data sets. We don't have enough time to cover every single data set that we've loaded. We've loaded over, I think, 35 data sets for this release, but we will cover some of them and highlight them in the various resources. In addition, we will cover some tools and functionalities that are already available to you and have been available for a while, but it's a nice way to remind everybody of what's available, what's possible within EuPathDB. And finally, we will also cover throughout the webinar some of the new features that we have. In particular, we will highlight the new genome browser that we've integrated. It's been available for a while as a beta version. So the genome browser is JBrowse. It's basically the newer version of the genome browser that we have used historically, which was called gbrowse. And the historic browser will be deprecated or not used anymore after release 46. So after October, we will not be supporting that genome browser, mainly because there's no support from the genome browser development community, and so it's becoming much harder to maintain. JBrowse is missing some features, but those are coming along. There is active development, and so we look forward to continuing updating JBrowse with new features and functionalities. The second feature we will highlight is a new RNA sequence summary page or summary view on gene pages, and I will show that at some point during the webinar. So for the first part, we will cover HostDB and some new data sets there, and for that, I'm going to switch to Eve, who will be covering that aspect. So Eve, I'm going to make you the presenter right now, and you can take it away.
Thanks, Omar. Let's see, so you should be able to see HostDB main landing page. Let me know if, yes.
Yeah, we can.
All right, sounds good. So I'll just make it a little bit larger so everybody can see. So welcome. As you noticed, I am on the HostDB main page, and HostDB databases essentially aims to integrate data sets relevant to host response to various infections. And today I will navigate to the new section, as Amar showed you before for the EuPathDB sites. But here we are on the new section for HostDB. And one of the data sets that we have integrated in this release is a transcriptomic data set that essentially consists of two data sets. One is for subpopulation of macrophages, mouse macrophages, and the second data set is essentially transcriptomic data sets of the pathogen used in this experiment, or Candida albicans. Here in this demo, I will take a look at the transcriptomic responses that were discovered in populations of macrophages that were infected with Candida albicans. And just sort of a brief overview of the subject, Candida albicans is a fungal pathogen that is quite important medically, as it forms persistent and sometimes drug-resistant infections in people with sort of decreased immune response, and also it's quite keen on forming biofilms, especially if people are on the long-term support using various medical instruments. So I will navigate to the datasets page by clicking on the link shown here. And that is essentially dataset record page that describes a relevant publication, as well as a brief introduction into what type of samples you are expecting to see, as well as the individual authors of the dataset presented here. And as we scroll down the page, you will notice that there is a few links that are offered. And these are essentially examples of the type of the searches that you can create using the dataset integrated here. I will show later a demo of transcriptomic investigation using a fold change option, but you can also set up a differential expression search or look for the genes, let's say in the 80th or 90th percentile of expression within the dataset. As you scroll down a little bit more, you will notice that there is a graph on the left, as well as description of the samples on the right. And so just to give you an idea of what we're going to look at just in a few seconds, I'll walk you through with the type of samples represented here. So the dataset have looked at the subpopulation of macrophages that encounter Candida albicans cells as a pathogen, and there were several outcomes observed, which translates essentially our samples. They were infected macrophages, which is AMO, stands for macrophage, that had live yeast cells identified, and there were 4 different samples within this subsamples rather within this main sample that are essentially different time points of collection post-infection. The second sample were infected macrophages that also housed dead Candida cells, and there's 2 types of time points, 2 and 4 hours. And the third sample that is represented here are actually macrophages that were exposed to Candida albicans cells, but they never became infected in such a way that live or dead cells were not identified within the cells. And there are 4 time points for this particular sample. And the fourth sample is essentially macrophages that were unexposed and are essentially your control. And in a demo, I would like to take unexposed macrophages as my control or a reference sample and identify genes that were upregulated in the infected macrophages that had dead Candida cells at 4 hours of collection. So let's go ahead and set up this search. I will navigate to the list of possible searches that you can set up and click on the fold change. And this essentially will redirect me to a main screen where you will be prompted to create essentially your own search of the data. Notice that you have a left and the right panel, and I'm interested to look at protein coding genes only, but you can also look at all genes if you wish, and mouse macrophages. I'm interested in identifying upregulated genes only at this point, but as you can see, you can have various choices available to you. Furthermore, I would like to select those genes that are upregulated by at least fourfold. As you can imagine, you can change this parameter to create more selective or less selective, so more stringent or less stringent searches. And here I'll move on into selecting actually my samples. My reference sample in this case will be the control that I have just mentioned, and these will be unexposed macrophages. So essentially the control that we're using, or the reference sample, will set up the reference expression level in your experiment, shown right here on the right. And next, you can select comparison sample, and in this case, I'm interested in looking for genes that are upregulated by fourfold in infected macrophages with dead Candida cells at 4 hours of collection. So here we are. As you notice that visual representation on the right have changed as well. So now you see that you're looking for the genes that are at least fourfold upregulated. So notice there's a parameter that's called floor that creates essentially a default setting at 10 reads. We have a help section available here that if you like, you can read more, but it essentially deals with FPKM values when they are close to 0. For example, fold change can be calculated in misleading ways if FPKM approach 0 in such a way that you can essentially get incorrect analysis of the FPKM data, and try to avoid representing misleading sort of output for the fold change. We raise very low FPKM values to default parameter, or essentially what is FPKM floor, which represents about 10 map reads as a default setting. This helps you to essentially avoid incorrect searches using the FPKM values represented here. Once we click on Get Answer, we will deploy our search, and the output will be essentially as shown here, where you would be viewing the results or a list of genes that fits your criteria. If we scroll down a little bit to the bottom, we'll see that now we have essentially over 1,000 of genes that map to expression values or fourfold or greater when we're using unexposed macrophages as control. Notice that you have gene IDs associated with product descriptions. You also have fold change numbers, as well as graphical representation within the column on the right. So you can technically scroll down and look, see if any genes or particular genes will pique your interest. Notice that the display on the right also demonstrate not only the results for the sample that we have chosen to look at, but at all samples across all of the experiment represented here. So that's a nice and quite useful visual representation of the data on the large sort of scale. However, imagine that you want to interrogate your data further, and let's say you may be working with a particular gene, and you simply want to know whether this gene actually is represented within the 1,000 of genes that have high expression values. So we know from the past experiments done by others that macrophages normally respond to various pathogens by increasing sort of expression of the inflammatory factors. And one of these factors is called chemokine, CXCL2, that is actually selectively expressed in-stress macrophages. So let's go ahead and set up a search for CXCL1. And if you're like me, I most of the time do not remember very long gene IDs. So we will use a text search to identify CXCL2 in mouse macrophage data set we're investigating here. So as you noticed, I clicked on the add step function to bring up an option for you to create a new search strategy. In this case, I'm interested in identifying gene using text because I do not remember a specific gene ID. And clicking on the text option will bring up a new search screen that essentially gives you a few parameters that you can either select all, or in this case, I will limit my selection to mouse as I am searching a mouse-related data set. I will also type CXCL2 in the text term. And since I am confident that this is the name of my gene, I'll just say search for CXCL2 and the gene name field only, and intersect this search with the previous step to make sure that you will return only the gene of interest and nothing else. And I'll click on Run Step. I have pre-run this step just before we started, so I will navigate to the screen now, and that's essentially the output from the search strategy that I created. And voila, the chemokine actually is present within my dataset. And as you scroll down, you will see that there are several transcripts available. So once you click on the gene ID link within this table, you will be redirected to the gene record page for chemokine CXCL2. From this point, you can investigate further the specific data available for this gene, and notice that we have a few options available on the left, where you can, for example, click on the literature section, orthology synteny, or you can look also at a gene model and to see what is known about the gene. Notice that here's, I clicked on the gene models and I'm redirected to essentially a preview of gene model in JBrowse. And as Amar noticed, mentioned before, we're transitioning into JBrowse. And here's a very quick look of the JBrowse representation of this gene. So if you click on the button here, you're essentially redirected to the JBrowse that I just pre-run. And it's quite a responsive, clean view. And in addition to viewing gene models, you can also activate various tracks. If you click on the Select Tracks button on the left, you will open essentially a selection screen where you can browse the various types of the tracks available and activate those that you're interested in. For example, I chose to activate those related to this particular data set, such as transcriptional analysis of the subpopulation of macrophages. And this is what I have chosen to view. Notice that this particular gene has different types of expression at various time points, which potentially indicate that these may be different splicing results in different points of collection, I believe, during the infection. So at this point, I will conclude the demo of HostDB, and I will give it back to you, Mark, but feel free to ask any questions that you may have or email us.
Okay, thank you, Eve. Quickly before I talk about CryptoDB, one thing I wanted to note about HostDB is that the goal, again, is to integrate data, as Eve mentioned, data from various pathogens that infect hosts and to integrate the actual host response to these pathogens. And so if I navigate to the transcriptomics section and look at the RNA sequence evidence section, this will list all the available data sets currently in HostDB. And so the one that Eve demoed was the one on Candida albicans, but obviously there are host responses from various hosts to the various pathogens that are available. And of course, this will hopefully expand over the coming months and years. One nice feature, obviously, of this is that you can also start asking questions across these various host response data sets. And while Eve was demoing, I was just curious, and I ran a search, which looks fairly complicated, but it's actually quite simple. I asked for, in the first one, I asked for all genes that are upregulated in the dead macrophage infected cells, similar to what Eve ran. Then I asked for any gene that's upregulated in a live infection. And I basically asked for anything that's upregulated in a live infection, but not in the dead infection. Presumably, this is an active host response to live fungi as opposed to dead ones. And then I was wondering, is there any correlation between upregulation of genes in different infection models? And so I intersected the results of this fungal infection model in mouse macrophages with an infection model of human macrophages with Leishmania major, in this case. And I had to transform by orthology from this experiment, which was done in human cells. So the result of this experiment is human genes. The result of the fungi experiment is mouse genes. And so I transformed the human genes to mouse genes by orthology and asked how many of these are in common between these two types of experiments. And the result came up with 9 genes. So, of course, I did not delve into these to determine whether these actually make sense biologically, but that's the kind of type of discovery searches you can do to identify potential interesting correlations between host response to various pathogenic organisms. So I'd like to switch and talk quickly about CryptoDB. So CryptoDB, for this release, we loaded a new data set that actually just got published in Nature Microbiology last week. As is common, in case you were not aware, but most people end up telling us about data sets they're working on well before publication, which is quite nice and allows us to really allocate effort and schedule the data set appropriately. And so in this case, we were able to schedule it to a release that was close to publication date and kind of is a nice compendium to the paper itself. As Eve indicated, if you click on the dataset itself, you'll be taken to the dataset description page or record page, which then shows you information about this dataset, including information about the experiment. In this case, this is a pretty cool experiment where, if you're familiar with Cryptosporidium, getting access to the entire life cycle has been really a big challenge, and work from Boris Strepin's group and a number of other groups has actually allowed completing the full life cycle, both in a combination of in vivo and vitro cell culture models. And so in this experiment, they had samples from various time points, including female gametocytes, sporozoites, oocysts, and a couple of time points for all of these. Again, the search, I won't delve into it in too much detail because Eve showed you the search in fungi, which is exactly the same search that will be available to you here. Just to highlight that from these different searches, depending on the type of experiment, you may or may not have all of these searches available to you. In this case, you'll notice that there's a fold change search, which is the search that Eve ran, but there's also a differential expression search. This allows, and the reason why we have this search available is because this experiment had biological replicates, so it allows us to reanalyze the data using the statistical power, so using something like DESeq2. And so this allows us then to compare samples to each other. So for example, I can ask for anything that is upregulated in the female in vitro culture compared to the asexuals. I can ask for upregulated. I'll keep it at twofold. I could keep the p-value as is or change it to something more stringent if I want, and then click on Get Answer. And again, this will result in giving you a list of genes that met the criteria that you were looking for. I could go back and click on Edit and Revise and say, well, maybe I should get something that's a bit more stringent. So let's change this to 0.5. And again, this is very nice and exploratory because I'm running this exact search for the first time, and the result may have been 0, right? And so, but that's what you end up discovering by running these searches. A couple of quick features before I switch back to Eve, where she'll describe some data sets in FungiDB. you have access to a number of tools once you run a search, including a quick ability to run a word cloud on the results. And this allows you to quickly see if there's anything interesting in your data set. And so you could see that the word kinase appears multiple times. If I mouse over the word kinase, it'll give me a number. So there are 51 occurrences of the word kinase and so forth. Of course, this is not a statistical representation of the results. It's simply counting the number of times a particular word appears in the name of the gene or the description of the gene. If I wanted to do something a bit more with more statistical power, I can analyze my results by clicking on the Analyze Results tab and then select from one of the analysis options that are available. For example, one popular one is running a gene ontology enrichment on your results. And so by clicking on this, I can ask for, is my data set of 985 genes that are upregulated in female gametocytes enriched for any particular Go terms? And in this, you can pick one of the Go ontology groupings. In this case, I'll just keep it a cellular component and submit this. This will run the analysis and will provide you with a list of Go terms that were enriched with a p-value, and so you can take a look at them and see if anything passes the threshold that you are interested in, and so forth. Okay, so I will stop here for CryptoDB, and Eve, I'm going to switch back to you so you can cover some of the aspects of FungiDB. One main thing is to, obviously we're cognizant of time, we're going to try and end this webinar in half an hour so we don't take too much of your time, but of course we may run a little bit over, but hopefully we can stick to time. So Eve, you will be presenter right now, and go ahead.
All right, sounds good. Thanks, Samar. All right, I am now on FungiDB, and that's the new section for FungiDB release. Notice that we have quite a few data sets integrated, ranging from the new genome sequence annotation project or a new sequence for non-pathogenic species closely related to coccidioides, which is the cause of the valley fever disease in humans and animals. Various transcriptomics datasets, SNPs, proteomics, DepSeq, and also updated phenotype records for Cryptococcus neoformans. So I will go ahead and show you a three-step strategy for starting essentially with a transcriptomic datasets for Cryptococcus. Notice also here's the second portion of the macrophages infected with Candida albicans infection that I have discussed for HostDB. So I won't go through details of how to set up a transcriptomic search, as you have already seen this. I'm now on the dataset record page. Notice I can scroll down and essentially see what type of searches are available. And this particular dataset actually looked at the wild type and in a CUF1 deletion strain and see how they behaved in response to various treatments, where it includes the copper deficient media and copper rich media. So this dataset was integrated from this publication here. And the reason why it's kind of a cool dataset is because we know that copper is important when it comes to fungal establishment and dissemination of the infection. For example, successful ability to deal with copper by Cryptococcus neoformans essentially translates in its success in being able to colonize tissues and internal organs. So instead of transcriptomics fold chain search, in this case, I was interested to look at the genes that were highly upregulated in the wild type treated with copper sulfate as a part of the medium, to look for those transcription factors that respond to increase in copper concentration within the environment. And so this is a sample of how you would set up a search. Essentially, you would say, all right, I want to look at this particular sample, and I want to identify all genes that are within from the 80th to 100th percentile in my sample. And essentially, once you click on Get Answer, you will arrive to a screen that would look something similar like that. In this case, I have identified that over 1,500 genes corresponded to my inquiry, having a high level of expression. But next, I wanted to actually ask a few additional questions to try to figure out what type of sort of interesting things I can find. Sorry. Here we are. One of the things, imagine you could be a researcher working with kinases, and we know that kinases are quite often upregulated in response to environmental stimuli. And in this particular case, I set up another text search. In this case, I have used kinase, and I have used asterisk on both sides to essentially look for any gene product that has kinase in its name. Sorry, in the gene product description. And this particular data set is mapped to Cryptococcus neoformans, H99 strain. So I limited the search in the organism to H99 strain only. This search returned over 30 genes that corresponded to my criteria. And the next step that I wanted to see is how many of those genes have actually phenotype associated either with mutations or with some sort of abnormal phenotypes reported in the literature. And this is where I have deployed an additional search by adding a step, and I'll just briefly walk you through how I set this up. I clicked on that step and navigated to our phenotype records that we have recently updated for Cryptococcus and clicked on phenotype evidence. So notice that you are redirected to another screen to set up your search. And the particular data set that I have used in my search is the Cryptococcus phenotypes data set. And I wanted to look at the curated phenotype records, CP. So I clicked on that. And the next step, I selected an organism in which I am, for which I am looking to see evidence. And in this case, I used two parameters to identify the phenotype that I was interested. First, I used the phenotype qualifier. And second, I used the specific phenotype that I wanted to identify. So I will log out from the screen and show you specific selections that I have made. For example, for my phenotype qualifier, I have chosen to look for those mutations that have led to the decreased resistance to a particular drug that can be used to treat fungal infections, such as rapamycin. So overall, I'm interested to look for the genes that have high upregulation in response to copper, yet those that are reported to have a decreased resistance to the treatment with rapamycin when the gene is actually either mutated or deleted. And once I set up my search, I was able to return that only one gene out of the 4 genes that have the phenotype associated with them, and that is the AGC-AKT protein kinase. So this is essentially, in a nutshell, of how you can start essentially interrogating data sets, for example, using transcriptomics data, and expand your data set further by combining and adding steps that essentially can utilize any of the listed data sets here. Okay. At this point, I will conclude the demo of the FungiDB dataset, and back to you, Amar.
Okay, thank you, Eve. So the next section will be to cover some new data in GiardiaDB. There was a question about which type of experiment to use, whether to use a microarray or RNA sequence experiment, especially when they have the same time points represented. I will try and demo this when I cover PlasmoDB, but the point is really all about how stringent or relaxed you want to be in your searches. So you can combine results from multiple different technologies and experiments and intersect the results, and that will be very stringent. Or you can use a union operation, which we can demo that as well, and that will sort of cast a wider net, and you can say, well, I don't care, as long as it's upregulated in a microarray experiment and an RNA sequence experiment, I want the gene, then that would be casting a wider net. So I will try and demo that a bit later. Suzanne, I'm going to switch to you to cover Giardia DB, so I'm making you the presenter. Go ahead.
Okay, sounds great. Thanks, Omar. So here we are at the Giardia DB homepage, and I'm going to go to the news, and that will show us that we have two new data sets. One is an annotated genome from the Stefan Sfard's group, Giardia muris, strain Roberts-Thompson. And this is a mouse parasite and the first Giardia genome sequenced outside of intestinalis. So if I go to that record page, we'll see that there's a long list of searches that are available for this, and that's because, you know, as we integrate all of the genomes, we run several genome analyses on that to predict domains and orthology, and then many searches are based on the annotation too. So one of them I'd like to show. So if I go up here to new search for genes based on sequence analysis and the protein motif search. There was a recent publication in 2018 about membrane-associated proteins in Giardia lambda. And one of those, I'm going back to the search page now, one of those motifs was FXNPXY. So so if I search for that, I can look for that in Giardia muris. And I can use the organism parameter to reduce the number of genomes that I search for this and click at answer. So we find that only 5 genes within Giardia muris have this peptide. That's a pretty useful search when you're looking for things. And then the other data set that we integrated is a proteomics data set. Let me go back to, and here we are at that data set record page. It was published in 2018 in GigaScience by the Aaron Jex lab. And it's about metronidazole resistance in Giardia. So metronidazole is a frontline drug treatment for Giardia, but treatment often fails due to resistance. Resistance is rarely genetically fixed, and lines can revert to sensitivity. So that's something really interesting that people need to study. And this study compared the proteomes of 3 metronidazole resistant lines with their sensitive parents. The data comes to us as quantitative data, and the ratio of resistant to sensitive cell line protein abundance in each line. So if we want to search this data, we can do new search for genes based on proteomics and quantitative mass spec data. We have one data set, and I choose that. So there are actually, these are the 3 cell lines that were looked at, 106, 713, and WB. So if I look at, run the search for 106 data, looking for upregulated genes at a p-value of 0.05 or a fold difference of 1.3, I can click get answer, run the search, and I find 87 genes. But if I really want to interrogate this data, I can use this search again and see what is the intersection between genes defined in 106 and genes returned by one of the other strains. So I click Add Step and use this Add Step pop-up to navigate to the search again. And instead of running the data on running the search on 106 data, I'm going to run it on WB data. using the same parameters, upregulated, protein coding, and look for the intersection of these. So anything that's upregulated in 106 would also be upregulated in WB, right? And I find that 6 genes are at the intersection of these two. And just to demonstrate the strategy system, I can also add step and add any search that I'd like to this. So I can also actually look for whether any of these genes, have, for example, predicted signal peptides, which is data that we've accumulated because of our genome analyses when we bring in these. Okay, I think we're done with Giardia.
Okay, great. So let me switch back, and what I'm going to cover right now is some new data sets and features in PlasmoDB. So I'm looking at the PlasmoDB news section. There were 2 new data sets that we loaded. One was an RNA sequence experiment from Elena Gomez-Diaz and colleagues, where they looked at, again, various stages, in particular salivary gland sporozoids and midgut oocysts. And the other data set is a microarray data set, which it's actually a quite detailed transcriptomic of transcriptome of the gametocytes. I think they took time points every hour or every 2 hours. And this data comes from Marie-Lynn Buchholz. And again, any of these data sets, you can click on them and get additional information about the data set, and see what kind of time points, for example, are selected. These are figures that are coming straight from their paper. In this particular case, the paper is in bioRxiv, so it's not yet officially published in a main journal, which it will be soon. And again, as everybody else demonstrated so far, there are graphs on this record page, but also you can go in and run searches against this data. One type of search I want to highlight, that's a search that we make available for some transcriptomic data sets, and that requires a large number of samples because it uses Euclidean statistics, which require a large number of samples. And this is the similarity search. If I click on this search, this allows me to find genes that have a similar expression profile to my gene of input. So I can leave this default ID, gene ID here, but you can replace this with any gene ID of interest, so your favorite gene, for example, and ask to return any gene from this time course that have the same correlation or expression pattern of your gene. And this presumably would help you potentially identify things that are in the same either regulatory network or potentially have similar functions and so forth. So this result is returned 50 genes here. And if I scroll down, I can see the genes that are being returned. And I have 2 profiles here. One of them is the profile of my gene of interest, the gene ID that I entered. And then the second one is the profile of the returned gene. So all of these genes are coming back with a pretty high profile similarity. You can look at the graphs and see what they look like. And now you can potentially pick some genes that are co-regulated or have a similar expression pattern for additional discovery, research, and analysis. The question that was asked earlier about whether we should use RNA sequence or microarray, again, that it all depends on, first of all, what do you know about the experiment itself? So it's always good to go back to the paper and read about it and understand what the experiment is about. And then once you decide that, yes, this is an experiment you really want to query, you can query it. But then I find it always very useful to integrate many different types of experiments. So let's say I'm interested in finding gametocyte genes that are upregulated at a particular gametocyte stage, and I really want to be confident about the genes that I'm returning. Then the best thing to do is to run the search in multiple different experiments from different labs, which are available in the database, and intersect the results with each other. So just like Suzanne and Eve were showing you how you can intersect results, you can intersect results from a microarray experiment and an RNA sequence experiment, or from multiple RNA sequence experiments and multiple microarray experiments. And you can even then combine them with results from a proteomics experiment, right? And so that's the beauty of being able to use this strategy system. So in the interest of time, I'm going to switch to show you a gene page in PlasmoDB. So I'm looking at just randomly picked a gene. And in that gene page now, we have a section in the transcriptomic section of the gene page called RNA-seq transcription summary. And this is, as we get more and more RNA sequence data sets in the database, it becomes hard for people to say, well, which experiment should I look at? You know, which one is an interesting experiment? And so this particular representation is an attempt to give you a high-level summary of what your gene is doing in these different experiments. So looking at this, I see all the RNA sequence experiments that are available in PlasmoDB. Each line here represents one of these experiments, and the dots represent the samples or time points in that particular experiment. So the x-axis here is log2 FPKM values. And so we're looking at expression values over the time course or over the samples from the experiment. And you can quickly look and say, oh, it looks like this gene, whatever I'm looking at this gene, for this particular experiment, the female and male gametocyte transcriptome from Edwin Lassander's group, it looks like the dynamic range of expression between the time points is very low. So my gene is probably not doing anything interesting in this particular experiment. But if I look at some other experiments, I can see that some of the time points are distributed widely, and I can click on this and see if my mouse will cooperate. There you go. So now you'll see here that that time point 35 and time point 10 here, 10 hours post-infection, seem to have the widest differential in expression, but of course they're distributed throughout. And so you may decide, oh, okay, this is an experiment that I really want to explore further. And so as you scroll down here, the transcript expression section will include graphs and more details about each of these experiments. So in this particular case, this experiment was from Ton Hake, and so I can go down now to this table here and look for that experiment. As you start typing, it'll filter the results, and here's the Ton Hake experiment. And so I can open this, and now this will take a few seconds. These graphs are loading dynamically, and so I can look at the expression profile, and you can see indeed here's the 10-hour time point, here's the 35-hour time point. That's the biggest differential, and you can look at more details of the experiment, including the coverage, which will load right here in a few seconds, showing you what the expression looks like. Note that there's an interesting warning here that tells me this gene has 9 paralogs, and so that means that the gene is potentially a part of a gene family. And so you have to be careful when you look at that with RNA sequence, because the mapping of the reads may not be mapped equally across the different paralogs, or it's obviously some reads will be thrown out because they've mapped to multiple locations in the actual expression analysis. Okay, so I'm going to switch back now to Suzanne, who will cover ToxoDB. And of course, keep asking questions if you have questions throughout the webinar, if anything is not clear or anything requires additional explanation.
Okay, thanks. So here we are at the ToxoDB news release page, and we see that ToxoDB got 2 data sets this build, an annotated genome and a genome browser track of non-coding RNA. So the annotated genome is a cyclospora genome, and that comes to us from the Food and Drug Administration, Dr. Gopinath at the Food and Drug Administration. And this is an organism involved in foodborne outbreaks. The genome was sequenced with Illumina technologies to about a 20-fold coverage. And again, without going into any detail, we have a host of searches that are available for that. This genome also includes the apicoplast and mitochondrial genomes, and that we can see when we search for genomic sequences for this. Sorry, I'm having.
Yeah, sometimes that type ahead doesn't work as efficiently as you would hope. But
right, yeah. So here I am. I've searched for all the genomic sequences that are associated with the genome of this new Cyclospora genome that we've integrated. There are 740 sequences, so it's not assembled into chromosome at the chromosome level yet, but there are, you can see that 738 contigs, 1 apicoplast genome, and the mitochondrial genome is here too. It's just not necessarily listed in this table, and that would be one of these two sequences. So
another place, Suzanne, if you weren't going to demo this, just to point out that you can also do a gene search based on genomic location. And once you do that, Cyclospora shows up there, and you can choose apicoplast chromosome or mitochondrial chromosome and just return a list of all the genes from those particular chromosomes.
Right. Okay, so I'm doing that now. Sequence ID.
No, go back to the home page, and let's maybe show it from the beginning just so everyone can see what. Yeah, and go to genomic location, and now select non-nuclear, the second one.
Ah, I'm sorry. I thought I had clicked on that. Okay, and that's the first one that we have, so we can get the genes from either the apicoplast or the mitochondrial chromosome, and click Get Answer. 92 genes. Great. So if there aren't any questions on that, I see there is one question. Perhaps someone else can look at that while I demo, because we're running out of time, the non-coding RNA. Yep. So the non-coding RNA we have integrated this time is data from the UPenn labs. It's available in our genome browser. So if we go to the gbrowse, so what they did for this data set is they isolated RNA from Giardia tachyzoites from several different time points, and then performed strand-specific sequencing on small non-coding RNA libraries. And here we are in JBrowse, which is soon to be our only genome browser. And I have 3 tracks turned on: the annotation track for ME49, and then 2 tracks associated with small non-coding RNAs. We got 2 data sets from them, one of tachyzoites from ME49 and one from tachyzoites of RH species or strain, and we've aligned both of those to ME49. There's a couple things you can do. You can actually look for, you can zoom in and look at these tracks a little harder, and so this might be an area that you're interested in where there are a lot more small non-coding RNAs aligned. And then you can also, you know, view details or zoom into this match or highlight this area here to further interrogate that. I think that's it.
Okay, I can switch back and quickly highlight something in TriTripDB, and then I think we will be potentially done. Obviously, we'll field any questions.
Sounds great.
Okay, so I'm going to just go to TriTripDB. So I'm looking in the news section in TriTripDB, and you can see that there are 2 types of data that were loaded for this release. One is a number of isolate sequences from G. congolense, and I will describe this in 1 second. And the other one is updated functional annotation. So we do have curators on staff who annotate particular genomes, and in this case, you can see the details of what has been loaded in terms of genome sequence updates or functional annotation updates. So we try and load these on a periodic basis from all the curators, both for Plasmodium, trypanosomatids, and various fungi. And as we get more and more of these, we obviously will and we have funding for additional curation, we will update other genomes as well. One thing to note before I actually go on to this particular query, if you'll notice here on the homepage, we have a user comment campaign going on. And if you're not familiar with these, this is a really great way for anybody to enter a comment on a gene page. And once you log in, you can go to your favorite gene and just say, oh, this gene is actually a kinase. It's published in this paper. I just read the paper, or I published it. Or you can even indicate information that you try to clone this gene, and the PCR primers that worked were the following PCR primers. These are all very useful comments that one can add on a gene record, and you are the owner of that comment. And once you click on submit comment, it appears automatically on gene pages, and it's searchable. You can even find these. So for example, I can find all genes that have user comments. If I click on this and get answer, this will return a list of all the genes in TriTripDB that have a user comment. In this case, there are over 9,000 genes that have a user comment. I can go down to these and check one of these out. Let's see what they look like. I'm just going to pick the first one. I have no idea what the comment is on that gene. You'll notice here that, wow, this gene has 10 user comments, right? So lots of people had something to say about this gene. If I go to the section, you will notice here, these are all the comments that are available about this gene. These were all provided by Joe Smith. You can go in then and click on the comment ID of any of these, and this will take you to the comment page, which shows you the details of all the comments. And in addition to text, you can actually upload images and other items, papers or stuff like that associated with a gene. And so this is a very nice way to crowdsource curation, basically, in a very crude way. And so I would encourage all of you to go ahead and enter user comments, not only because we have a user comment campaign going on, but on a regular basis. During this user comment campaign, if you're attending the NPM meeting, come by our booth and pick up a mug for your user comment. If you're not, but you know somebody who's attending, enter a user comment and ask them to pick up a mug for you. So that would be totally fine as well. So quickly, I know we're at the end of the webinar, but I just want to make sure I highlight this population biology data set. So if I click on this, I can, again, as usual, take you to the summary page for this data set, which gives you more information, including a link to the publication. And more importantly, of course, all of these potential searches that are available to you. And I'm going to just take one of these searches. Let's just take, I'm going to identify genes based on SNP characteristics. And if I select this one, this will take me to the SNP page or SNP search page. And you'll notice here I can select from many organisms that are available to us that have resequencing data available. So in this case, I'll just keep it at T. congolensi, but you can choose to change this. And now I can go in and either select all of the isolates from the study and say, I don't care which isolate, I want to look for genes that have a high number of non-synonymous mutations, for example. Or you can zero in and select specific strains from the study. If you know something about the strains, of course, looking at the paper will help a lot. Or you can do it by any of the other information we have about these strains. So for example, you can go in and see that there are strains from various parts of Africa that may be worth investigating independently or together, depending on your interest. In my case, let's just select all of them. And I could go in and say, I'm looking for all genes that have, let's just pick non-synonymous mutations. There are multiple criteria here that you can select. I won't go through them in detail right now. We may want to dedicate a full webinar just on population biology data, but I'll just mention that there is a lot of help information if you mouse over these question marks about each of these parameters. In addition, there's a number, or actually not on this page, but so the most of the help will come from these help parameters. The frequency basically tells you something about the number of reads aligned. So in a haploid, if you're looking for a haploid SNP, then you'll be looking for something that's 80% or higher. So of course, you never get 100% of the reads aligning or the reads matching that because you have errors in the sequencing. But if you're looking for diploid SNPs, you can go down below 40% and say, well, as long as 40% of my reads, or give me SNPs that where 40% of the reads have or show a particular polymorphism at that position. So let's just keep it, let's look for a haploid SNP. And I can say that out of all of these isolates that I selected, I want at least let's make this 80%. I want at least 80% of them to have SNP calls at the positions that are being considered to be returned, because sometimes you can have good sequencing in a bunch of the isolates, but then a bunch of the other isolates in your study may have bad sequencing in that, or sequencing reads in that particular area, and you potentially don't want to consider those SNPs. You don't even want to look at them if half of your population doesn't even have reads that match to that area. So that depends. This just increases the stringency of your search. So I can ask for anything, any gene that has non-synonymous SNPs that are haploid, not hap, but that are homozygous, SNPs in this case. This is a diploid organism, and I can run this search. Again, I don't know what the answer is. The answer may be 0, or it may be a high number. So that's the nice thing about being able to edit these searches, that you can go back and modify your search results based on your interest. And so this returned 2,301 genes that met my criteria. Scrolling down, you could look at them. You can see the total number of SNPs per gene, and the number of non-synonymous SNPs. And so you can see this HEC domain protein is highly, in theory, a highly polymorphic gene with many non-synonymous mutations. Of course, you'd have to delve into these a bit more detail before making any broad conclusions. Okay, so I think this covers everything that we wanted to discuss during this webinar. Eve and Suzanne, anything else or any parting points?
Not for me.
Okay.
I don't have any.
Okay, great. And are there any additional questions? We'll stay on for maybe another 10 seconds or so to see if anybody is fielding any questions. And again, as usual, if you think of a question later on and you think, oh, shoot, I should have asked that question during the webinar, don't worry, just click on the Contact Us link on any of our websites, and one of us, Eve, Suzanne, or myself, will answer the email, and we try and be fairly quick in responding to emails. So feel free to use these. We love to hear from you all the time. And of course, if you notice any issues in the website, also let us know, because we are not looking at every single page on the website all the time, and so it's good to hear from you about suggestions for improvements, and also alerts for any kind of bugs that appear on the website. Okay, so with that, I think we'll go ahead and end the webinar. Thank you very much. And as usual, this webinar has been recorded and will be made available to you afterwards, and also put on our YouTube channel for anybody else to view and examine. Thanks a lot, and have a great day. Bye-bye.